start.
Okay. Oh, great then. All right. Okay, so we'll get started. 1 second. Okay. So welcome everyone to our next Malaysian Research Insider session in 2021. I hope everyone is staying safe and healthy, and I hope some of you are watching the badminton match, which I also am on a side screen. Let's really pray and hope that our Malaysian team wins. But let's get on with our event for today as well. So to those who are new to the Research Insider session, allow me to quickly brief you about who we are and what this session is. So this live stream is streamed on the MBIOS Facebook page, as well as the Science Media Center Malaysia Facebook. We provide a chance for everybody to discover our talented Malaysian bioscience researchers, and this would be an amazing platform for the general population to have an opportunity to interact with them. So each scientist that we host here will discuss their work, their experiences in science, and also the details on how they have gotten to where they are right now. So these conversations will be accessible to all of you and are directed at non-specialists. And the general format of this is that the speaker starts with an overview of themselves and their topic of conversation, followed by a short Q&A session with questions from all of you. If you're interested in or learning about topics outside of your area of expertise, please don't forget to tune in every third Saturday of the month, prepared to chat with a Malaysian scientist and to learn something new. I'll start by quickly introducing myself. I am Ashwini Ilangovan. I'm a PhD student in biomedical sciences at the University of Pittsburgh. This science chat is organized by MBIOS and media partnered by 100 Scientists of Malaysia and Science Media Center Malaysia. And a quick reminder to our audience, please feel free to post any questions that you have in the chat box at any point during the talk. All right, I'll move on to introducing our speaker tonight. Our speaker tonight is Dr. Kavita S Subramaniam. Dr. Kavita is a research associate at the King's College London. She works on addressing the unmet needs of a rare skin condition called Recessive Dystrophic Epidermolysis Bullosa, RDEB, also known as the butterfly disease, by utilizing a patient-friendly gene therapy delivery method to better improve their wound healing process. Without further ado, let's welcome our speaker for tonight, Dr. Kavita. You have the stage.
Thank you, Ashwini. Hello, everyone. Firstly, a big thank you to you, Ashwini, and the MBIOS team for doing a grand job organizing this Research Insider Talks. It's really impressive the way they've set up everything from first contacting the speaker and then to make sure everything will run smoothly. Well done. Secondly, a personal thank you to the audience here today for either spending your dinner time or lunchtime listening to my talk. It's a real pleasure to have this platform to chat with all of you. So I've been asked to share a little bit on my background, my research, and my day-to-day life as a scientist. I'll walk you through each of these soon. And at the end, please feel free to ask any questions. So I come from Tamping, a small town in southern Malaysia, here highlighted in yellow, which borders Malacca and Negeri Sembilan. And I know that there are many Tamping people in the audience today, so shout out to everyone. I do miss my hometown dearly. I took my entire educational journey from kindergarten to my PhD in Malaysia, something that many people have been curious about since my early schooling days were in schools that were in the outskirts rather than in big city. All I can say is that we had really good teachers who pushed us to our potential. Perhaps the first flavor in science that remains in my memory was when I was in Standard 3 with my primary school science teacher, Cikgu Jeffrey. I'm sure many of you would have had the same task where you're asked to grow bean sprout seeds in various conditions with or without water, in the sunlight or in the dark. And the next day when I saw the results, obviously seeds with water and sunlight grew compared to the others. It was like a light bulb switch moment for me, that to get conclusive answer, you need to test in various conditions. So it's a solid observation, which then equated to a scientific thought process. My secondary school, in my secondary school being in science stream, biology was my favorite subject opposed to maths or physics. So I knew I would be ending up doing something in the science field for my future, but I did not know exactly what would I be doing. So after my SPM or O-level, there were lots of questions whether, do you want to do medicine? Do you want to do pharmacy? I was really interested in forensic science, but I was informed that in Malaysia, without formal police training, you can't go through forensic science training. Then I just wanted to do something in science, basically. So this brings me to my first question to the audience. Is being in your current field what you wanted to be when you were young? I know we have a diverse amount of audience here today in various different fields. So I'm curious to know, is being in your current field what you wanted to be when you were young? It's a very interesting outcome. Should we close it in 10 seconds? 5 more people. Okay, so 53% of you answered yes, and 47 of you answered no. So that's a very close call. And in my case, my initial ambition was to be a researcher, any researcher, but on the moon. Not entirely sure how to set up a research facility on the moon, but I was quite adamant about it for many years, until after high school when logic set in, and I enrolled for a Foundation in Science program on Earth. And at that time, I was made aware of this relatively new course by my mom, which was called bioinformatics. And when I visited the open day, not really knowing what the course entails, I was given reference to the TV program CSI and how bioinformatics is a combination of science, maths, and programming. It was really intriguing. And at that time, it was a leap of faith for me because it was a very new field. And we were the second batch in MMU to undertake that course. But I'm glad because that sort of pushed me to know where my strengths were at that time. Throughout the 3 years of my degree and various exposure to programming and biology, I could already sense that I was leaning towards the biology side of the course. Miss Ong, my mentor, shown here in the photo, was a big influencer for me to cultivate my love towards research. And then I realized during my final year internship, when I was in my final year, I did an internship at this research facility purely on bioinformatics, that I needed more variety and challenges in a lab setting. So the programming aspect of the course was not giving that to me. So I then discussed with Ms. Ong, and she helped design my final year project to be an entirely lab-based project. I got to work on type 2 diabetes and got to experience firsthand the sweet success and the frustration that goes in research, but I loved it. So after that, after 3 years, and I finished, graduated my bachelor's, I was at a crossroad again. What am I supposed to do next? One would say the next logical step is to join the workforce. But my gut said that I'm not done with science yet, and I would like to learn more. But in which direction do you go? Do you do more research? Do you do master's by research, master's by coursework? Do I still want to continue researching about type 2 diabetes, or shall I try out another field? So then at that point, I started sending out feelers to various university lecturers, inquiring for a research assistant position in Malaysia. If I were to like it, I could always register for a master's. And if I did not like being in research, I can then think about what else can I do. At that time, many did not respond to the email. Many declined or rejected. And I am aware that this is a familiar situation amongst many postgraduates here. But I kept on trying and trying until, by chance, I was put in touch with someone who ended up being my supervisor for 5 years. I had heard from Dr. Ivy Chung, and at that time, she was also a newly appointed professor at the Faculty of Medicine in UM. Together, we wanted to start a project on pancreatic cancer in UM, which was actually a continuation of her own project from Boston. So, through twist and turn, in the end, we were contacted by a gynecologist at PPUM who wanted to collaborate with us to research on endometrial cancer, which ended up being my thesis project for about 5 years. It was a long but fruitful journey. We got the grant, we had the samples, good science was done, and still being done by this group, and I'm really proud to say that. We had pressure to publish, we had fun at conferences. Essentially, my PhD research was to look at endometrial cancer cells and its friend in the tumor microenvironment. One such friend is what we call the fibroblast cells. We identified that the cancer fibroblast cells secretes factors that helps the endometrial cells grow more than what it would do by itself. So 2 publications resulted from this work. And as you can notice here, I've put in a cheeky statement mentioning that I converted from a master's to a PhD. There is a difference in time scale, but I mentioned in the beginning of the slide that I had no idea or planned to pursue a PhD. I only spoke about masters. But as I got more involved in the project, there were more aspects to research about. And with encouragement from my supervisor and the availability of research grant, I went for a PhD conversion. Perhaps the most poignant takeaway message that I would like to share is that to be open to any changes along the way when you are doing your postgraduate studies. You may plan to study one disease and end up doing something totally different. You might have not planned to stay for too long and then ending up staying for 5 years. But think of it as an investment for your skills and expertise. Often when you're doing a postgraduate study, people often think that you are a student and you don't have any job experience. I realized how very wrong this thought is, as during a postgrad is the time that you're collecting experience, sharpening your skills, learning all the skills that you would eventually be using as long as you're in science, whether you're a student or not. So then after that, in 2015, a memorable experience came out of my PhD. It was an opportunity to do an internship with a well-known pharmaceutical company, Novartis. It was a tough competition. There were about 500 applicants worldwide, and they were only choosing 20 people in total. I'm really grateful to have been chosen, and it gave me a taste of what it is like to be working in a pharma industry. Whilst I did not do any lab work there, I was working on the clinical trials that my supervisor, Olga and Panos, were working on. And I came up with a clinical trial design, learning what I had learned from Novartis, implementing it to my research. Should there be an endometrial cancer clinical trial happening in Malaysia? Additionally, I was also very lucky to meet so many good scientists from around the world doing excellent work in their own field and a chance to learn from them. We had remained in touch over the years, and it was a very rewarding 3 months. One thing I would say is if I could go back in time, I would have done more internship or more attachment, lab attachment, helping out my lecturers and tutors during my sem breaks. Even 1 to 2 weeks of being in the lab, it is a high value experience that is immense, and it propels your CV to a much, much higher level. And then after I got a taste of what it is like working in a pharma company, I came back, I finished up my thesis, graduated from It was a really memorable moment. And then, of course, I was at a crossroads again. I was really tempted to go into the industry thanks to the brilliant experience at Novartis. Speaking with my supervisor, she was supportive, but I guess she knew me much better, having worked together for about 5 years, as she really told me that think if I'm actually done or ready to leave bench type research, as if I were to go into clinical trial, it might be very long before I get the chance to hold a pipette again. So I was thinking, and then I decided that I still want to be in research, as I don't think I'm done with it yet. So I started applying for jobs. I don't think there were any days that I did not apply for jobs right after I got my submission approved for PhD. So it was a long process. Lots of emails sent out, lots of feelers sent out. Basically, all you do is ask whether they have a position, and then you look at job adverts, you apply for it, you customize the cover letter. It's a lot of effort, but if you think it's worth it, it will be worth it. And my second question to the audience would be, I know there are people in industry at the moment and university in the audience at the moment. What would you do? Would you stay in academia, or would you move to industry, or would you swap? Okay, so we have we have more than half of you saying that you will move to industry. So thank you for the feedback. If I was answering this question 6 years ago, I would also have chosen the second option. But years make you know yourself better, and I am glad that I stuck with option one. So then I got an offer from Imperial College London. It was a big step. Not only the fact that I'm moving countries, but it was also a new field altogether, and I didn't really know anyone in London at that time. And making friends as an adult, it's much more difficult than making friends than when you were a kid. So it was a new environment. There's a lot of excitement. First job, it's a new project. I was tasked to carry out a project to study normal brain function. So it was a new challenge because I wasn't studying a disease, but I was studying how normal body function goes about. And then I realized soon enough that, as much as I love learning about new condition or new biology, that loving the project alone is not sufficient. There needs to be a synergy between your boss and the way that they see the project progressing. There were lots of issues, some of it solved, some of it did not get solved. So I decided towards the end of my probation that it was really not worth my effort and stress level that I was having at that time. So then, of course, it was a bittersweet moment. I was in a country that is not home. What do I do next? And start back to the same process of job application, sending out emails, and all that all over again. Something that I just did about 6 months before this job. However, there are some lessons learned. If the group has a high turnover, and this is something that's important that I would like to highlight, there must be a reason for it. And as much as you can investigate your potential new boss, ask students, ask previous lab leavers, some things are just not meant to be. And my dad always says that when one door closes, the other one opens. And the other door that opened was something that I never dreamt of. It was an offer from the University of Oxford, and surprise, surprise, I was working in the gynecology field again. Essentially, the team and I were working on uterine fibroids, and the hypothesis was that there are factors released by fibroids that causes changes to the blood vessels in the uterus to behave abnormally, and this leads to heavy menstrual bleeding. And this relates to patients, as portions of fibroid patients suffer from heavy menstrual bleeding. So from this project for about 2 and a bit years, I had the experience of collaborating with a pharma company, again, biopharmaceuticals. And apart from the usual academic progression, I also learned about patents, publication restriction, which I did not experience in academia before, but also how this patent, when filed early enough, can benefit your research as it's backed by a pharma company. The way that pharmaceutical company approaches their research is very different from how academicians would approach research. So then I was offered to extend for 2 more years, and I loved Oxford, I loved the group, I loved to stay, but an even better offer came by, and it was really exciting for me to explore that, and it would be a regret if I had not taken that opportunity. And that offer came from St. John's Institute of Dermatology in King's College, London, to work on a disease called RDEB. We'll talk about that in a few minutes. But let me just take a few moments to introduce the institute, as there might be medical students on the call thinking about derm specialization in the future. So St. John's has been around for a while now, since 1868, at the time ran by volunteer GPs. The institute moved to its current position in Guy's Hospital in 2007. If you've seen London skyline photos, which I'm sure you have, is the building right beside Shard here in this white arrow. And the institute works on various skin-related conditions, but mainly these four: genetic-related condition, photobiology, immunodermatology, such as psoriasis, atopic dermatitis, eczema, as well as oncology. For my project, I'm in collaboration with an international team and a biotech company, which I'll explain shortly. And the study is funded by a charity called DEBRA. One thing to note is that I was really pleased that I was given the confidence to lead the work from September 2019. And I was offered the position, although I did not have any dermatology background at the time. The important outcome that was highlighted by my bosses who interviewed me at that time was that I had the skills that are needed for the job. The skills, if you remember me mentioning, has been from accumulation throughout the postgraduate years. So you really don't know how in the end things all tie in together. So my next question to the audience would be, have you heard about RDEB before? RDEB stands for Recessive Dystrophic Epidermolysis Bullosa. Thank you. 18 of you answered, and all 18 of you answered no, which is exactly what I would have said prior to September 2019. I did not know about this condition before I took up my current job. In fact, skin is something that I've always taken for granted, but not anymore, and here's why. So RDEB is a condition of a bigger umbrella term called EB, epidermolysis bullosa. Perhaps the most relatable example to mention when I speak about EB is this touching documentary called The Boy Whose Skin Fell Off. It is the real-life story of Johnny Kennedy suffering from this condition following his final few weeks of life. He unfortunately passed away during the filming of this documentary, and I would encourage the audience today to find this documentary online to gain a better understanding of what EB patients go through. So with regards to definition, EB is the name for a group of inherited skin disorder that causes the skin to become really, really fragile. That's why that's the relation of the butterfly children here. And the classification depends mainly on where the blisters form in the layers of the skin. So we know that our skin layers are called epidermis and dermis. If the blister forms in the epidermis layer, it's called simplex EB. If it forms in the dermis layer, it's called DEB, dystrophic EB. And if it forms in the middle of these two layers, it's called junctional EB. And in my research, I'm working mostly on RDEB, which is the recessive form of DEB. And some facts and figures about RDEB, which is a subset of EB. There's an incidence of 3 cases per million live birth and a prevalence of 1.35 cases per million population. It is unfortunately hereditary, so it's genetic related, and it is autosomal recessive, meaning each parent can carry a copy of mutant gene, but they don't show the signs or symptoms, but the child would when they inherit the dysfunctional gene. So it is the most severe form of EB, and the blisters are present all over the body, not just on the skin, but also the mucous membrane, such as the moist lining of the mouth, digestive tract as well. And as the blisters heal, as they might do a bit slowly, they would result in severe scarring. And scarring on the skin would be fine to an extent, but scarring in the mouth and esophagus can make it really difficult to chew and swallow food, leading to chronic malnutrition and slow growth in children with EB. It is very painful. It leads to very poor quality of life, and there's always a risk of progression to squamous cell carcinoma over here. The cause of it is due to mutation in collagen 7A1 gene. This gene is on chromosome 3 of the human genome. It results in few or none anchoring fibrils. Anchoring fibrils are those that are highlighted here in green. And you can see here in healthy skin how anchoring fibrils holds the dermis and the epidermis layer together. It's like a Velcro that holds both layers together. But in RDEB skin, because there's no anchoring fibrils, there is nothing to hold it together. Therefore, the slightest friction or trauma can cause blister, as shown by photo here. These are real life photos of patients that has come through the institute, and it is very painful. So how about in Malaysia? I came across this article from October 2019. I could not find any facts specifically for RDB in Malaysia, but its parent group EB has about 60 people registered with the Malaysian version of the charity that I'm funded by, DEBRA Malaysia. And it is estimated that there's about 150 to 300 Malaysians that have EB and suffers from this condition. So then one might ask, what has been done about this awful condition and what is the cure? The fact that collagen 7A1 gene is the cause for this disease was only discovered about 30 years ago. It's as young as me. So from the 90s to now, many various angles have been tried and tested in terms of therapy. Unfortunately, there's no single curative treatment for this condition yet. So researchers have tried injecting whole collagen 7A1 gene, have tried to make skin equivalents, and more recently, we have opted to go through the route of gene therapy. So gene therapy is different from gene editing. Gene therapy and gene editing are two most recent efforts that has been done in the field of EB or RDEB. It has been around since the 70s, but it's only been used for EB and RDEB from the early 2000s. To put simply, gene therapy allows the cell from patient with no collagen 7A1 gene to be manipulated to express collagen 7A1 gene so that anchoring fibrils can be produced to hold the dermis and epidermis layers together. So you transfer a working copy of the gene into specific cells of an individual. You can use either viral vector or non-viral vector. Gene editing, on the other hand, sounds exactly as it is, and it has progressed tremendously in the past decade from using CRISPR-Cas9, where you introduce double-stranded break in both copy of your DNA, to prime editing, where you only need to break one strand of the DNA, to base editing, which is literally like Word document search and replace. You search one base of the gene, of the DNA that's causing mutation, and you replace it. So these are very beneficial to EB and RDEB because we know what gene causes this disease. We know in which area in the gene there are hotspots, and we know if we remove the defective gene and replace it with a functional one, the disease can be reversed. So let me ask you now, have you heard about gene therapy before? Either it does not necessarily be in RDEB, but any other disease or any other circumstances. Okay, we have a mixture of results actually. 52% of you say yes and 48 say no, which is great because it is new and it is there to be utilized. How do we harness it? That is another question. So then let me then ask you, from the press, either good or bad press that gene therapy has received over the past years, do you think gene therapy is ethically wrong? So choices are yes, it's wrong, no, it's not wrong, and the third one needs careful implementation. Perfect. Okay, so 71% of you said needs careful implementation. 29% said no, it's not wrong. Good to hear. And no one said yes, it's wrong, which is good. So, you know, it's undeniable how gene therapy can be beneficial in the following success stories, such as there has been contribution of gene therapy in treating hemophilia, in immune system disorder, sickle cell disease, as well as reversing blindness. In terms of EB, one major success story from 2016, 15 to 16, was to create a new skin to replace what was caused by EB on a 10-year-old boy. And such story gives an encouragement for researchers in EB to continue to harness the advantages of gene therapy. And from my review, in terms of gene therapy, these are the active clinical trials currently ongoing for gene therapy. And one such trial that I would like to highlight, and this is in relation with the previous slide, as well as what the work that I'm doing at the moment, is called EB-101, where a biopsy is taken from the patient which has RDEB, who has RDEB. You isolate the skin. We have keratinocyte and we have fibroblasts from the same skin layer. You expand, you transfer the correct form of collagen 7A1 into the cell. And then the corrected keratinocyte would then further be expanded to form cell sheet. And this cell sheet will then be ready for application. Usually, it will have to be grafted onto the wound bed. So imagine a patient who's already going through a lot of pain, and the only option they have is a new skin sheet. It has to go on operation, through an operation method. Whilst that might not be their initial choice, they are willing to do that, and they're willing to go through that just to have a chance at a normal life. So in relation to my project, there are two main aspects. Firstly, similar to what I've shown previously, that is to perform gene therapy and to carry the correct form of collagen 7A1 gene. Secondly, with patients' pain and ongoing suffering in mind, instead of grafting or injecting, which is invasive and it causes more pain, we have collaborated with a biotech company to instead spray the gene-corrected cells onto patients. So RenovaCare, the company that manufactures these spray guns, have been in the skin industry for a while now, but they have been working predominantly successfully with burns patients, a patient who had some sort of third-degree burn, second-degree burn wound. They have taken skin from unaffected skin from other parts of their body and then sprayed it onto the burn wound, and they have reversed the scarring. So let me just walk you through briefly about the first part of my project. So to illustrate, the idea is to have a virus vector carrying the correct collagen 7A1 gene. This is done using an age-old technique called restriction digest, where you cut the vector and you put in the functional collagen 7A1 gene in, and then you streak them on an agar plate. The next day, if indeed the functional collagen 7A1 gene has gone into the vector, the backbone, you will get various colonies. You'll get all these white dots here are called colonies. Each colony carries one functional collagen 7A1 gene. You'll then pick the colony and you'll grow them up and you'll extract the DNA to validate if it is indeed the right one. So the idea is to have a vector having all this additive to make the collagen 7A1 gene functional and expresses well, and then to bring them into patient. Then once you have the vector ready, it's time to take the skin samples from patient and transduce them so that it picks up the functional gene in and return it back to the patient using the non-invasive spray, skin gun spray. So this is the vision that we have. And in terms of just a little bit to illustrate on the results that I get from the lab, as I said, our skin layer is epidermis and dermis. From the epidermis, you get what we call the keratinocyte cells. These are cells that I'm growing in the lab. And from the dermis layers, you get the fibroblast cells. And when cultured together, they form a little bit like little islands with fibroblasts flanking the keratinocytes, mimicking our actual skin layer. Now, when we talk about spraying gene-corrected cells back onto patients, there are a huge number of considerations to think about. So, do we spray fibroblasts alone? Do we spray keratinocytes alone? Together? Does it matter if it's mixed? And hold on, the fibroblasts and keratinocytes used, they grow in different nutrients. They have different media to grow. How are we to overcome that? And then, of course, there's the ratio conundrum, where do you spray it in a 1-to-1 ratio, fibroblast to keratinocyte 1-to-1? Do you do 2-to-1? Do you do 1-to-2? Which one? Which one do you go ahead before you even think about bringing it to patient? So this is something I like to highlight, and this sort of relates to what I thought when earlier when I spoke about those bean sprout seed experiment, that you have to think through everything just to answer one question: which cell-to-cell ratio works best? And from my experiment and from my observation, when we show one slide containing all the results, there's huge amount of work that had gone through in the background. So to make the claim that what can go to patient is a 1-to-2 ratio in keratinocytes media only, and it doesn't matter which cell goes on first, you need to do a lot of different validation work before even coming to the conclusion. And how do we, in the group, envision us taking this to the patient? As I mentioned earlier, we are halfway there. We have the gene therapy technology, and now we need to do as many testing as possible using the spray device before bringing it to patients. It is worth noting that EB is a rare disease. So the authorization to use the spray gun or any sort of technology in patient would not go through the same clinical trial route, for example, what the COVID vaccine had gone through. It will be usually done through a compassionate use basis, where the treating physician sees the patient, if or not they would tolerate the treatment well, and then suggest that they go through whatever technology or whatever trials that are in progress at the moment. So we're approaching the end of my talk now, and I was asked to say a few words on how my lab life looks like. So those of us in research know that often we plan something, and instead of a straight route, you have ups and downs, and you spend a lot of time troubleshooting. The downs are the one that propels you to up most of the time, and this is important to remember when you're planning for an experiment. Also, whatever results are shown, this is exactly what I said earlier, is usually just the tip of the iceberg. And when it's a success story, there's a huge amount of effort that went into making the experiment results even worth mentioning. I've also realized over the years that I would like nothing more than to come into the lab, do my experiment, analyze my results, and go home. But the longer you are in the field, the more additional work comes through your way, and this is something that's unavoidable. So now, in addition to my actual experiment planning, analysis, troubleshooting, I'm also expected at the stage of the career that I'm in right now to supervise students, evaluate papers, getting back to emails, and there's so many admin paperwork, and it is a balance that you need to strike in your day-to-day work. My final question to the audience: after this brief talk, do you feel that you have a better understanding of RDEB? Perfect, brilliant. Thank you so much. I am pleased to see this number. 100% of you said yes. And lastly, a few words of acknowledgement. Organizers, again, fantastic job. Keep spreading STEM awareness in Malaysia. 100 Scientists of Malaysia as well, who has featured my story a few times before. Wonderful people that I met throughout the years, and I'm sure many more to come. People from MMU, people from Novartis, friends in Oxford, friends in King's, friends outside of work who have helped me when I needed them. And last but not least, my most important cheerleaders, my family, my pets, my partner, and his family. Being in science can sometimes make you feel lonely, and the workload can be overwhelming. But you need to ensure you have an anchor all the time. And these are my anchors, whenever and wherever I've been, to pull you from going down that rabbit hole. So thank you so much. And thank you, audience, for listening to my talk today.
That was a really, really great presentation. Thank you so much, Dr. Kavita. It was really nice to listen to your story, as well as the different paths that you had to take to get to where you are now. And I'm sure this is not the end. There is definitely more to come. And I'll just start by saying that we are really proud of you and hope you continue the amazing work that you are doing. So with that, I would start with a few questions that we have so far. So we have a little quirky one. How does it feel to be offered a job from Oxford University?
Well, I did not expect that. That's the easiest answer I can tell. I remember going for the interview after my experience with Imperial. I went for the interview in the morning, took the train to Oxford, had the interview, came back, didn't come back. I was still hanging around in Oxford, visiting a museum, and I got the call to say that I've been offered the job, whether or not I want to take it. So it was a bit surreal at that time, and I called my parents immediately and said, I got the job. So it was it was a proud moment, but at the same time, it was also surreal.
As I would expect it to be, because it is Oxford. Okay, another one. And I completely echo this question. So it starts by saying you're amazing, which again, I said earlier, this is a really nice story for all of us to hear. And yeah, again, we're really proud of you. But what is your motivation? What keeps you going every day?
So I would say that I feel like I'm lucky because I'm in a field that I'm really enjoying every day. Sometimes people say, today's Monday, I need to go to work, but I don't have that feeling because of the job that I'm in. It's not just a job, it's a project itself. I think it's a combination of having really supportive team members, bosses that have confidence in you, and your interest in progressing the project as well. So every morning when I wake up, I feel excited to go into work rather than having to think, oh, another day, another job, another experiment. No, it's never like that at all. My motivation is actually to see this current project to completion, because I know it has so much potential, and I know that the patient translation isn't that far off. And King's, especially St. John's, being St. John's, they have the capabilities to do compassionate use with a strong case. So I know that this will reach patients probably in the next 5 years. So that is a big motivation to make sure that what I'm doing in the lab now reaches the patient as soon as possible.
Absolutely. Okay, so the next one is specifically about gene therapy. So they're asking, what is your view on how to educate people more about gene therapy? And this is really important because you know how people are already, some people are terrified about even taking the vaccine, right? So if you tell people that, you know, we're doing gene therapy and we're going to be inserting virus into you, how would people react to that? And how can we as scientists do a better job at educating them?
I think it's important to highlight the plight that the patients undergo, because if we're not even going through half of, not even anything what the patient is going through. For example, skin is something, it's always just been there. It's sort of like part of the furniture almost, that you can't even think someone having to think about wearing a different clothes, wearing a different, not falling off a bike, not making sure they don't injure themselves in any other way. I mean, I'm talking specifically about RDEB, of course, but if you can show people what patients go through and then show them the potential of what gene therapy can help in this patient, I doubt that they can see that any differently. I think what is important is, what does the patient benefit from this? It's not about whether you or not you agree when you're not going through that condition. It's what the patient benefits.
Yes, totally. Okay. Okay, somebody asked, what is your advice for those who would like to pursue a research career but lack lab or research skills or just the experience?
Experience is gained through time. I mean, when I started in my final year project, I didn't have any experience either. All I had was my undergraduate lab sessions of 3 or 6 hours every week. for the past 3 years. So you gain this throughout time. It is something that you accumulate and you gain. But what I would say is, if you are really keen and if you are interested, scientists and researchers are always open to having an extra set of hand to help them in their work. So, ask your tutors, ask your lecturers, because they might be doing, if you're in a university setting, you would be doing some sort of research as a lecturer or a tutor, and ask them whether they need any help. Is there any possibility to maybe assist you 1 to 2 times a week? Or, do you need me to do DNA extraction? Can you teach me how to do DNA extraction? Be curious, be busybody, because otherwise you would not get the experience. And in the beginning it's fine, but as towards a certain point in your career, you would have accumulated because you've been a busybody before.
I can totally echo that. You have to ask to get the experience, and you will never know if you like something or not without getting to be a part of it. And a lot of us, we love science, but not necessarily we would love to do research. Research is science, of course, but there is a lot more to research than just liking science. So yes, anybody who is interested in getting into research, please do try and get as much experience as possible, no matter how small that could be.
Yes.
Okay, so back to gene therapy. Is it possible for gene therapy to be cheaper in the future?
Yes. So I'm saying yes, but it depends on the demand, of course. The skin equivalent story that I showed earlier for the 10-year-old boy, from inception to implementation, it took, if I'm not mistaken, it took about 30 million euros. That is one patient. So when asked, this is the same question that someone in my group asked the researcher, Michele De Luca. He asked us back, saying, what is the value that you can place on a patient that has recovered? Can you place a value on a patient that has recovered? So right now, it is expensive. It's not cheap. Depends on the disease, of course, but if a rare disease such as EB or RDEB can benefit from, various different funders and various different organizations that can make it cheaper for the patient, then sure, it can be cheaper in the future. But at least in the UK, what I'm aware of is, if such gene therapy comes into circulation, through compassionate use, patients can get the benefit of it. So the NHS or the health services would be absorbing the fund to implement the gene therapy towards a certain patient.
We all hope that would be the case in the future with more research, of course. Okay, now a personal question, I guess. Where do you see yourself in maybe 5 to 10 years from now? What are your personal future goals?
Well, this is the same question that's been asked in every interview as well. So
I'm sure.
I always say that, you have to have a plan A, plan B, and plan C. Plan A is right now in my immediate future, I would be seeing this current project to completion. And when I finish this project, depending on opportunities and depending on availabilities of research grant, whether or not the project needs me as much as I need it, then I would like to continue being in the project. But if other opportunities come by, then you'll always have to be open to accepting those opportunities, whether or not it's in research or whether or not it's in pharma. Eventually, I think there's a statistic saying that only 2% of academics end up becoming a tenured professor, tenured researcher. We don't know who's going to be the 2%, but all you can do for the moment is to do as much as you can: publication, research, presentation, good science.
Very nice. Okay, so we have a question asking, would it be difficult to get a job in the UK, both as a postdoc in the academia or in the industry? And do you have any tips on how to go about this?
So I have to say, because it is, when you do apply for a job outside of Malaysia, you do have to, you have extra considerations such as visa, documentation, and a lot of it costs money. So I would say, first of all, make sure your finances are stable. I was lucky because I had backup of my father and mother association. So, they were there for me financially at that time. And secondly, is it hard or is it not hard? I think it's subjective, depending on your field, I guess, and how niche your field is. I would say that if you think you can contribute to the project and to the sciences of the project, sign out as many applications as possible. I mean, I have a folder full of application at that time, and I've lost count of how many applications I sent out. And if you're the right person for the project, they will contact you back. In terms of working in the UK, I would say make sure that your documentations are all in place, especially, you would need an IELTS qualification, that you need to have your English level at a certain certificate in order for them to consider it. You need to pay for your visa, you need to pay for the health services. So it's money as well as documentation. But think through it in an organized way. It is a lot of information. It can be overwhelming, but think through it in an organized way. If you really do want it, go for it, apply for the job, and then do what you need to do to make sure that your job application, you accept the offer, and then all the documentation can come through with that. We all have experienced that with various things. Even in Malaysia, when you apply for a PhD or a master's, you have to go through health checks, and there are 5 different documents you need to fill out 5 different times, and it's the same, just one step a little bit more complicated because you are a migrant going into another country to work.
Very true. Okay, and then more so on your postdoc side of experience, were you required to learn new skills and techniques that you hadn't already acquired during your graduate studies experience, or were you already expected to know most of the skills that are required for the group and for their specific type of work that they do?
No, so I get asked this a lot as well. So do you need to know everything before you start your job? No, you need to know certain things, because that's why you applied for the job in the first place, because you have a certain base level skill set. Things that you don't know, be open to learning, and be a quick learner as well. There are no restrictions or qualms in people teaching you skills over here. If you don't know something, go find someone who knows it and ask them to teach that to you. And by the time you know it, you also know the technique. So no, you don't need to know everything.
Right, that was great. Okay, I think that was
there's a question here from Zoom. Can you see it? Is someone from a bachelor's?
Oh yeah, I can't see her.
Hold on. I was wondering if it was because the mutated gene is the one we are targeting. Yes. Can I get back to this question personally to Lindsay Yi, because I have a little bit more scientific backing up to do?
Sure. Or Ziyi, is that her name?
Yeah.
Stay on the call for a little bit. We will have a quick 5-minute casual chat with our speaker. So if you could just stay on, that would be great too. We had one more question.
Yeah, I was in a dilemma which path to carry on after SPM. However, watching a slice, better vision, what I like to go with. To SPM leavers, tips for SPM leavers. I was just like you guys in 2007, dinosaur ages ago. But I think you need to think whether you want to be which field you want to go into, because I know many of my peers at that time, even though they were from science stream, they wanted to go into something completely different, and they've been thriving in that field. So do you want to continue to be in science? Then you need to do some science related courses. But I know there's a conundrum there, and there's a dilemma there, especially when you've just left school in an organized structural learning, and you're thinking about what to do next. And I would say that, trust your gut feeling, whether or not you want to be in science or you don't want to be in science. If you don't want to be in science, don't force it, because you would end up not liking it down the road. pre-U and pre-matriculation and all that is a requirement anyway. So you still have a few more years to go before the major decision-making factor comes in. So yeah, I mean, there's nothing else I can say except to trust what you want to do, Britisha.
Perfect. I think that was the last of our questions. Yeah. Okay. So yes, thank you very much, Dr. Kavita, for the very informative presentation as well as the Q&A session. And that brings us to the end of the session today. Thank you everyone so much for your attention through both Zoom and our Facebook Live. And if you joined us late today, don't worry. You can watch the edited video on our MBIOS YouTube page within the week. We will also be posting up announcements on our social media once it's up. We will be taking a break from the Research Insider sessions in August to allow for maximum participation for our MBIOS Symposium, which will be happening the last weekend of August. Please keep an eye out for more information on that. We do have our advertisements out for our poster competition, so please do take a look on our social media. If you are already doing some research or you already did some, this would be a great opportunity to just put yourself out there, just to be able to present to people. Especially with COVID, we don't have too many conferences or meetings to attend. So any opportunities that come your way, just take it up. Again, experience is all you want, right?
Yes.
Yes. So we will see you all again on the third Saturday of September for our next talk of the year. And with that, also, I'd like to quickly highlight, we won a bronze medal in the men's doubles badminton. So yay, Malaysia. And yeah, so everyone, thank you so much for attending, and good night. And for everybody on the Zoom call, please stay on. We'll have a very quick casual conversation with our speaker, and we'll call it with that. All right, so thank you, everyone.